Yeah, so thank you for the invitation to talk. I know most folks in the room here. For those of you that don't know me and some folks online, I'm Dan Coleman. I'm an assistant research professor in the microbiology and cell biology department. I help advise Lisa Keller, who's the real star of the show that we'll be hearing from later on today. And so the charge was to kind of give you an idea of some of the research that's going on in the lab. It's sort of an introduction to the research that Lisa is going to be talking about as part of her dissertation work. So I'll just jump right into it. Slide advancer doesn't work, so I'll use these things, hopefully. So one of the foundational questions that we're interested in our lab is trying to understand what the nature of early life was on Earth. So asking questions like, where are the places that life might have originated on Earth? And what were some of the characteristics of early life, some of its physiological characteristics? Right, and so a couple things worth noting right off the bat is that we have a pretty good idea that life's been around for 3.8 billion years, very shortly after the formation of the Earth. And the Earth was really a microbial world for the first couple billion years, right? So life was around at 3.8 billion years ago. We have good fossil evidence of life at about 3.5 billion years. Multicellular eukaryotes didn't really arise until about 2 billion years ago. So for this big chunk of time, it was mostly a single-celled microbial world. The Earth was very different at this time. A lot hotter for volcanic activity. Oxygen wasn't really widely available until about 2.3 billion years ago. And so when we think about the types of environments that life was originating in, very different than the familiar Earth that we think about today. In fact, some of the best evidence that we have for early life on Earth comes from the Dresser Formation in Australia. There are fossilized microbial mats in this formation dated to about 3.5 billion years ago. You can see this in this hand sample picture here, right? These little wrinkly structures. Lots of work done on these geologically over the last several decades. So what we have a good idea about now is the environmental setting where these microbial mats were arising in were likely very similar to subaerial hydrothermal systems. So in other words, hot springs, perhaps not unlike the types of hot springs that we see in Yellowstone today. Right? So see these microbialites growing in the alpha of this geyser here. And so the environment that supported some of the earliest life on Earth was hydrothermal, and this is one of the reasons that we go to these types of hydrothermal systems today, hot springs like in Yellowstone, right? One of the reasons I, as well as others, go to these places is to try to understand something about the physiology of the earliest life on Earth by looking at the types of life that live in these really geochemically heterogeneous environments. Hopefully these pictures impress upon you the types of heterogeneity you can find in hot springs. So it's worth asking the question, though, that are contemporary hot springs actually really reflective of an early Earth? I mentioned a few slides ago that we know that Earth's chemistry has changed quite a bit. Most notably, the level of oxygen that's in our atmosphere through time. And so we think about life that arose 3.8 billion years ago. It would have been at a time when it was largely anoxic on early Earth, very little oxidants available for microbial metabolism. So what I'm showing here is just a schematic showing our evidence for the partial pressure of oxygen in the atmosphere as a function of present atmospheric levels, 10 to the 0. So this is today. Partial pressure is what it is today in the atmosphere, going back through time 4.4 billion years, right? And so when life was first evolving 3.8 billion years ago, very little oxygen. It really didn't accumulate appreciably until about 2.3 billion years ago during the Great Oxidation Event due to the onset of oxygenic photosynthesis and the activity of photosynthesizers. So there are consequences here, not just for the availability of oxygen, but in Earth's redox chemistry in general as life is first getting its foothold. And so these are some of the questions that we're interested in. We've been funded through a NASA project for the last handful of years, both Lisa and I, as well as Eric Boyd, collaborators at Montana Tech through the NASA EPSCoR program. And this is the question we're interested in asking. So are contemporary hot springs really reflective of an early Earth, knowing these things that I just mentioned to you about Earth's chemistry and how it's changed over time? So thinking about it in a little bit of a different phrasing, so can we look at relationships among geologic setting, geochemical variation in hot springs in these settings, contemporary microbial community ecology in these places, and their evolutionary histories of the thermophiles living in these hot springs, in order to understand how reflective these environments really are of early Earth? And doing this in two very different geologic settings for reasons that I'm really not going to go into detail for brevity in this talk, but looking at a place that's a little bit more reminiscent of early Earth, like a basaltic setting in Iceland, versus a place that's maybe less reminiscent of early Earth, like the rhyolitic setting in Yellowstone today. Right, and so our general approach for doing these studies is to look at all this geochemical variation that we see in hot springs, just showing a couple pictures here, very different colors, very different geochemistry in these springs. Understanding how that influences the microbial communities that we see in those systems, so what types of geochemical driving factors influence the contemporary ecology of them. Integrating that into a phylogenetic framework so we can understand how the evolutionary histories of these contemporary organisms may reflect changes in Earth's history over time. We can then trace back and try to understand something about why we see communities the way they are today and as they relate to geochemical variation. Right, so this is our sort of general approach. I'm going to talk a little bit about some of the data from this overarching NASA-funded project to lead into LISAs. A couple important things to know about hot springs before I jump into it. So one of the first things that you'll notice if you're going around hot springs across the world, whether in Yellowstone, New Zealand, Iceland, or wherever, is that you're going to find a bimodal distribution of pH, right? So a distribution that looks a little something like this graph here. Lots of acidic springs between pH 2 to 4, lots of circumneutral alkaline springs between maybe 6.5 to 8.5 or 9, right? With very few springs in the middle of that. So why is that? Still doesn't work. It's kind of a habit to use this clicker, but it's not actually connected to the presentation. In case you see me waving this around, just trying to get it to change. So our current model for hot spring formation, this is the same in Yellowstone as it is in Iceland and anywhere else in the world, is that it starts with meteoric recharge, rainfall, snowmelt, percolates downwards, interacts with the subsurface hydrothermal water reservoir, thought in many cases to be briny, so very ion rich. As the water circulating in the deep subsurface, it gains heat. It also gains volcanic gases, most importantly, sulfur dioxide gas. And so what happens is sulfur dioxide disproportionates when it interacts with water, forming hydrogen sulfide, a gas, as well as sulfate, an ion. So this water now has a lot of gas, has a lot of ions. It's coming up to the surface, and it undergoes a process called decompressional boiling, right? So as it's going up to the surface, pressure differences cause it to boil, right? And so you can kind of think of this as being analogous to like opening a soda can, right? So when you pop the top on your Dr. Pepper or Mountain Dew or whatever you like to drink, you're releasing the pressure and you're causing gas to come out of solution, right? So you now have a gas phase and a liquid phase in your soda. Similar sort of analogous thing happens in the deep subsurface in places like Yellowstone. So you get gases, more liquid phase solution. So those liquid phases can go onto the surface, carries all those ions. A gas phase, and this is really kind of the important part, can interact with near surface waters that carry oxygen. And so when those waters interact with those gases, principally hydrogen sulfide, be oxidized, we get sulfuric acid, right? And so this is where we get acidic springs in Yellowstone and other places, where we get circumneutral springs. It's this basic geophysical process that's similar across the world. And so the important point that I want to impress upon you about this whole scheme is an important player in the broad spring geochemical variation that you find across the world. It begs the question whether or not these processes would have been similar on early Earth as they are today, right? And this is what we're interested in trying to answer. Again, still doesn't work. Maybe it will eventually. So talk a little bit about the actual data from this project to lead into some of the stuff Lisa's going to talk about. We just wanted to ask the question, do hot spring microbial evolutionary histories and the functions that they encode vary within different types of springs? I'm going to talk mostly about the Yellowstone work. Lisa's going to talk a little bit more about the results from both Iceland and Yellowstone. In essence, we just used that approach that I mentioned a few slides ago. Right? Integrated microbial phylogenetics. We looked at community ecology parameters, taxonomic and functional diversity, and geochemical variation to try to triangulate all of these types of data and get at our question of interest. To do this, we crisscrossed the park, sampled 37 different high temperature hot springs over 60 degrees Celsius, sediment communities, as well as fluids for geochemical analysis. As shown in this map here, these are the different points that we sampled. 11 different geyser basins, over 60 different geochemical analytes, listed a few here. It was exhaustive, right? We characterized as much of the geochemistry as we could. Tried to do this across the range of temperature and pH that you find in hot springs in Yellowstone, as well as the different types of hydrothermal waters, which I'm not going to go into detail here just for brevity and to not take you into the weeds too much. So that's what we did. For our microbiology side of the analyses, we use a culture-independent environmental genomics approach. Many of you are familiar with this. You probably do these things in the lab. For those that aren't, just to briefly kind of orient you to where this data comes from, we go to natural communities. We can isolate cells. We can isolate DNA from those cells, the genomes from those cells, sequence it, and then through various bioinformatic methods, essentially stitch those genomes back together bioinformatically into bins, or what I'll refer to as metagenome assembled genomes, or MAGs, as folks call them. And so then from those genomes, we can infer community ecology metrics, phylogenomic analysis to get at evolutionary histories, as well as metabolic and biogeochemical functions. And so this is what we did for our giant data set, generated over 1,000 different MAGs for it. We clustered these into species level units so that way we could compare distributions in MAG or species across different spring sets. Ultimately came up with about 350 different species level units. Just to give you an idea of whether or not we did a good job of exhaustively surveying the genomic diversity in Yellowstone, I'm showing a rarefaction curve here. And the way to read that is that on the y-axis is just the unique number of species units that we find as we sample more and more of these MAGs. And so the idea would be that, like, as you sample more genomic diversity, eventually you're going to run out of unique species level diversity that you're finding, suggesting that you basically found everything, right? And so that's what you'd want to see if you comprehensively sampled something. As the figure is showing you, we're starting to level off there, and so we think we did a pretty good job at capturing all the genomic diversity in the system. At a broad level, how are microbial species units distributed across these different springs? Give you the answer right now. It's pH is what controls everything, as many of you have guessed. This figure is showing a non-metric multidimensional scaling plot. Many of you probably know how to read these. Just briefly, for those that may not be familiar with them, each one of those points is a different metagenomic community. So there's 37 of them for all the springs that we sampled. And the points that are closer together in space have more similar compositions in community. The points that are farther away in space have more different compositions in community, right? And so closer together, more similar. I've overlaid on each one of these points the pH that the spring came from, and you can see just visually that things cluster by pH, right? The blues cluster with the blues, the reds with the reds, and so forth. If you do a statistical analysis, pH comes out as the most important factor that affects community composition among all of the geochemical analytes we looked at, right? Still doesn't work. And so why is that? Well, the basic idea is that these species level units, as I call them here, are distributed across hot springs based off of their pH niches. This is a network analysis showing this data in that kind of way, where each one of these different boxes is a different species level unit. So there's 350 of them. The boxes are connected to each other when there's a significant co-occurrence in these two species across springs, right? So, you know, this species commonly occurred with this species here. And then again, I overlaid them by pH, their average pH of their distribution, to give you an idea of what their niches are, right? And so again, visually you can see blues cluster with the blues, greens with the greens, yellows with the yellows, right? So organisms generally co-occur in cohorts based off of their pH distributions. And so I'll leave that there for this part of it. Lisa's going to talk more about the network or these interactions among different species in her part of the talk. The question that we're really interested in asking then is what is the concordance of evolutionary history of these lineages with their contemporary ecology? And so to get at this, we conducted a phylogenomic analysis, took all 1,100 plus of these genomes, along with 1,600 others from all major divisions of archaea and bacteria, put it into a phylogenomic tree using 30 different universal housekeeping genes. This is a cartoon schematic of that analysis, or the product of that analysis, where all the red lines are the genomes from our data set in the context of all the major divisions of archaea and bacteria. Quite a bit of diversity, as you can see. The question, again, really interested in understanding is how evolutionary history is perhaps variable or associated with different spring geochemistry types. And so one way that I looked at this, or the primary way that I looked at this, is by looking at the distance of each one of those genomes to the root of the tree. Right? So the tree is rooted, the bifurcation between archaea and bacteria, that's where those two domains split, calculated all of the distances from each one of those branches to that root to get a function of how early branching or how deep branching they are, right? So the longer branches are going to be more recent because they're later diverging. Smaller branches are more closer to the root, more early diverging. You can consider them older, right? And so what does this data look like when we aggregate it across our entire data set? It looks like this. So what this is showing is the distribution of all these distances to the root for each one of these different genomes across all the genomes in the data set. And I've grouped them by metagenome. So there's 37 of these boxes here, and increasing pH from the most acidic all the way to the most alkaline in deep red there, right? And so those distributions show for that metagenome, all those genomes in that system and how far or close they are to the root of the tree. Again, visually, you can look at it, you can see that there's a pretty big difference with pH. It's not all over the place. There seems to be more early branching or older lineages in the mid pH springs relative to the lower pH and higher pH springs. Looking at this data in a little bit of a different way, you can see this very clearly when we look at the median for all of these different maps, right? The mid pH springs have the earliest branching lineages, pretty clearly. So this is kind of counterintuitive to me in thinking about this, right? The least abundant types of springs that we find in contemporary hydrothermal habitats, continental hydrothermal habitats, host the most basal branching lineages. I think maybe the reason it's counterintuitive to me because I would have expected like the most abundant springs would have the best analogs for early life. It doesn't seem to be the case here. And so for the last few slides that I'll show, one of the things we wanted to figure out is what physiologically about these organisms enables them to inhabit these particularly not abundant springs relative and be early branching relative to the other types that are more abundant in continental hydrothermal systems. So to do this, we reconstructed the physiological potentials for all 1,154 of these genomes to evaluate these physiological and evolutionary associations. This is a crazy plot that I'm not going to go into very much detail on. It's just to impress upon you. This is sort of what the data looks like in a large form, right? Each one of these rows is a different physiological function. Each one of these columns is a different metagenome. Circle sizes are just how prevalent that particular function is in that particular metagenome. Just looking at it, you can see that pH is important, right? The sizes of the circles in the low pH versus the mid pH versus the high pH springs. There's different distributions. If you do a statistical analysis, this bears out as well. And I'm just going to talk really briefly about each one of these different types of springs in turn, just to give you a feel for what's sort of causing these patterns, right? And so looking at acidic springs, I told you before, they form by oxygen-dependent sulfide and sulfur oxidation. They tend to harbor high sulfur content. You can see this in this picture. There's obviously a ton of sulfur in the spring. Lots of metal contents from leaching. They tend to be dominated by recently evolved lineages, and in particular archaea, like the sulfolobales. And these organisms primarily rely on aerobic oxidation of sulfur and iron compounds. And so overall, these springs are very oxygen dependent, right? They're reflecting oxygen-rich earth, both in how the springs form, but in also the organisms that dominate them. Turning to these circumnatural alkaline springs, also reflect an oxygen-rich earth. These tend to be very poor environments for organisms to inhabit. There's not a whole lot of gas in them. They tend to be energy substrate poor. They tend to be organic carbon poor, but they contain a lot of arsenic. And what do we find when we look at the organisms in there? We're dominated by relatively recently evolved facultatively aerobic bacteria and archaea. These organisms tend to be enriched in metabolisms like arsenite oxygen-dependent process, as well as arsenate reduction. And then we know from other work that we've collaborated with others on that arsenate in these springs is largely dependent due to oxygen-dependent oxidation of arsenite. Last thing that I'll mention about these springs is that from our other studies, we know that their waters are dominated by obligate aerobes, and these are probably only present in near surface waters due to atmospheric oxygen ingassing. This is some work that we published in Maria's paper, and some other understanding from Eric and myself. So these springs, perhaps not a great analog for early life either. Primarily, the ecology of them is dictated by oxygen-dependent processes that will harbor the deepest branching lineages. And the last set of springs that I'll talk to you about are these weakly acidic sort of mixed springs. They tend to be visibly black. So springs like this, that's because they have high levels of iron sulfide minerals. They also tend to have the lowest levels of oxygen, high levels of gas, and moderate substrates of energy, concentrations of energy substrates. In fact, as I mentioned before, they tend to harbor the most diverse and the deepest diverging archaeal and bacterial lineages, and these organisms are enriched in metabolisms that use gas substrates, so methanogens that produce methane, hydrogenotrophs that are essentially using hydrogen as an energy substrate, carbon fixation through the Wood-Lung-Bell pathway, which is thought to be an ancient pathway. All of these have been speculated to be features of early life on Earth. Right. And so these not very abundant springs in contemporary systems are kind of neat, right? So they harbor the deepest branching lineages, anaerobic functions, which we think contributes to why you find those sorts of organisms there. And so I'll leave off there for my part of the seminar. Just a couple key takeaway points. Hot spring geochemistry more or less reflects the contemporary Earth conditions that we find them in today. And the most tend to not be the best analogs for early Earth by these analyses. It's actually the relatively rare spring types, these weakly acidic mixed springs, that tend to harbor the deepest branching lineages with metabolisms reflective of early life characteristics. And I think these data have relevance for how we consider early life analogs for study and understanding how life has co-evolved with Earth over the last 4 billion years. And the last thing that I'll say is that these data are obviously from one system, from the Yellowstone system. These patterns might be a little bit different in different geologic contexts, and this is a question that we're actively working on figuring out. So that's it for my part. Hopefully I didn't eat up too much time into your part, Lisa. I guess we'll just hold our questions until the end. If anyone has them for me, we can do that, or you can catch me afterwards. So I'm going to turn it over to Lisa for her talk about her dissertation research. A couple things about Lisa. Many of you in the crowd already know. She's been a great contributor to the lab. She came to us in 2019 from North Carolina State University, where she worked with Bob Kelly, who's a big figure in microbiology, if you're not aware of the name, where she worked on thermophiles and model organisms like Sulfolobus and Pyrococcus and the like. And really where her love of extremophiles kind of began, right? And brought her here to Montana State to do a PhD with Eric and working on this NASA grant that I just mentioned. Lisa's been incredibly productive since she's been here. Has 8 papers already published in places like Nature Communications, PNAS, Applied Environmental Microbiology, et cetera, et cetera. Most notably, her most recent paper was just picked up by the New York Times a couple weeks ago, as well as the Boston Globe, I think BBC News, and who knows how many other outlets, which is great. So Lisa's obviously a rising star in the field, a great contributor to the lab. Yeah, and so with that, I'll go ahead and turn it over to you, Lisa.
Thank you, Dan, for that introductory presentation. That was really good introduction to what I'm going to be talking about. Thank you for introducing me as a speaker. As Dan mentioned, I'm a 5th-year PhD student in Eric Boyd's lab, and today I'm going to be talking to you all about cross-feeding between 2 dominant populations in Yellowstone and Icelandic hot springs. And so to start off, I want to give just a little bit of background information to kind of lead into what I'm going to be talking about. And so Earth contains many ecosystems with extremely unique community compositions. So when we look at planet Earth as a whole, there are countless different ecosystems within Earth. And here are 3 basic examples of those. So this top right picture is a high alpine ecosystem in Rocky Mountain National Park. The bottom left is showing a savanna, and then the bottom right is showing coral. And so just between these different ecosystems I'm showing, you can see that they look completely different. There's really no similarities between these except for that there's some plants, there's animals. But one thing that I really want to note within these ecosystems is that in a specific ecosystem, we have different community compositions. So if we're looking at like an alpine ecosystem, for example, different alpine ecosystems all have different community compositions. And so the big question is, why is this occurring? So in ecology, there's this idea about community assembly. And so we know through ecological studies that community assembly actually dictates the observed populations within an environment. And community assembly is a pretty complex topic, but this schematic does a really good job kind of breaking it down into little smaller portions that are easier to understand. And so I'm going to be talking about this in the context of hydrothermal systems or hot springs. So to start off, we have our regional hydrothermal species pool. So here for the WebEx people, I'm going to use my clicker to point this out. So we have our regional hydrothermal species pool. And so these are all of the organisms that could potentially inhabit a hot spring. And so these go through the first filter of dispersal, where organisms are taken from the species pool and spread out to different environments. And then after dispersal, these organisms go through an environmental filter and a biotic filter. And so this environmental filter is essentially abiotic influences that change or that impact what species or organisms can be in a system. So for example, in a hot spring, this would be something like pH or temperature or nutrient availability. So the next filter that they go through is biotic interactions. And so this is biologically mediated, something like competition, for example. And so once we go through those two filters, we end up with our local communities. And in hot springs, that would be, for example, an acidic hot spring, a moderately acidic hot spring, or an alkaline hot spring. And so when I focus in on alkaline hot springs, we have interactions that are occurring within this specific local community that kind of facilitates and ends up changing the communities that we have within that local community. And so that kind of essentially is the reason why if I go to one alkaline hot spring and look at the microbial community, and then go to another alkaline hot spring and look at the microbial community, they won't ever be exactly the same because of these interactions occurring within the hot spring. And so to kind of give an example of a macro ecology system, I want to look at the kind of food web of Yellowstone National Park. And so we know that resource availability and the interspecies interactions that are occurring significantly impact the community structure within an ecosystem like Yellowstone. And so one thing I want to point out is that primary producers in an ecosystem are extremely important. And so primary producers are organisms or plants or algae that can essentially generate their own energy and their own food source. And then that ends up feeding primary consumers. And so when you look at this food web, you see that there are a lot of interactions, and it's an extremely complex environment, an extremely complex system. And that is what ends up facilitating so much diversity within the animals and plants and everything that we find within Yellowstone National Park. And so in the Boyd Lab, being focused on hot springs, I'm kind of wondering, so what does this look like in a hot spring system? And so here we have a hot spring picture. Here's the source of the hot spring. And then here we have the outflow channel. And you can see that this has really vibrant green and orange colors in the outflow. And that's indicating that photosynthesis is present in this outflow. And so the way photosynthesis works is these organisms take carbon dioxide and they take light energy, so energy from sunlight, and chlorophyll, which is causing that pigment, the color that we see in that hot spring outflow, and they convert the carbon dioxide to glucose, which is then cross-feeding the primary consumers, or as we refer to in the microbial world, the heterotrophs of the environmental system. But one thing that I want to point out is that geochemistry can actually inhibit the presence of photosynthesis in hot spring environments. And so what I'm showing here is a diagram showing the presence and absence of photosynthesis, and all of these points are hot springs that have been sampled. And you can see that they're ranging on a wide pH and temperature distribution. And it's really clear that anything above pH 5 and above a temperature of 70 degrees Celsius, we have inhibition of photosynthesis, and anything below pH 5 and above 50 degrees Celsius, we again have complete inhibition of photosynthesis. And so in these environments, instead of relying on photosynthesis, they're actually supported by chemosynthesis for energy production. And so, like I mentioned, we have different aspects or different portions of a hot spring. And so in the source, this is where the hydrothermal fluid is originating from, where it's coming from in the hot springs. The source is oftentimes a lot hotter than the outflow of the hot spring. And oftentimes we have photosynthesis completely inhibited in the source of hot springs. And so instead, they're using chemosynthesis. And so to explain what chemosynthesis is, we have a really similar structure where organisms are using CO2 as well, but instead of using light energy, they're using chemical energy. So in this example, they're taking hydrogen sulfide and oxygen in order to convert that carbon dioxide to glucose. And then again, that glucose or other organic carbon forms end up cross-feeding the heterotrophs within the population. And so one really cool thing about this is that we can actually measure autotrophic activity levels within hot springs. And so we do this through the use of CO2. Previous work that was published in PNAS Nexus for my thesis research was that we sampled Old Faithful in order to determine whether or not there was microbial activity present in this system. This was a pretty big study because this was important study because this is the first time that high temperature geyser systems were investigated for the presence of life. And so what we did is I sampled 2 different water sources coming from Old Faithful. The first one here is the pool. You can see here in this picture, this is the opening of the Old Faithful geyser. So this is where the water is erupting out of. And I want to note that the only source of water in this pool is the water erupting out of Old Faithful. There isn't any other area or place that the water is coming from. And then we also sampled the vent water. So this is the water that's actually erupting out of the geyser. We caught it in the super high-tech plastic bin, and then I collected my samples from this bin and put them into serum vials. And then once I had my serum vials set up and we were back in the lab, we used a radioactive labeled CO2, and then we incubate these at whatever temperature you want. And for this experiment, we chose 70 degrees Celsius. And if activity present in these samples, we'll see an incorporation of that 14C labeled CO2, and they take that up and put that labeled carbon into their biomass, essentially labeling the biomass, allowing us to measure the level of activity within these samples. And so here's the data for that, but before I start, let me orient you all. So on the y-axis here, I have my activity level, and this is in counts per minute, or CPM. And so this is what we get off of the instrument that's measuring the activity. Then on the x-axis here, we have our pool water at 70 C and our vent water at 70 degrees Celsius. And we did these incubations over a course of 16 hours. And so when I show the data, you can see that there's a clear increase in activity over that 16-hour incubation period. And so this was actually really exciting, a really exciting finding, because this was the first indicator to show that there is active life within geyser systems. And so naturally, as a microbiologist, I want to know who's present, who's actually doing this activity. And so in order to identify this, Dan talked a little bit about this, but I'm just going to go through it really briefly. What I did is I collected biomass through this filtration system, and then I performed a DNA extraction on that biomass, and eventually that DNA was sequenced. And so the way you can think of DNA sequencing is that it's essentially like a genetic fingerprint of the cell, and it allows us to answer 2 basic questions of who is present in the system and what are they metabolically capable of doing within that system. And so using our sequencing data, we were able to identify that Thermocrinus was the dominant population in both the vent and the pool for the Old Faithful samples that we collected. And one thing that we also learned through our sequencing data was that Thermocrinus is an autotroph, and so it's able to take that CO2 and convert it into organic carbon. Another population that I want to talk about and point out is Thermus, and based on sequencing data, we identified that Thermus and all of the other organisms within the system were actually heterotrophic. And so they are not able to take that carbon dioxide and convert it to organic carbon, and instead are most likely relying on Thermocrinus in order to do that. And so with this data, we actually were really intrigued by the presence and dominance of Thermocrinus and Thermus together. And so that kind of made us wonder, is this relationship more common? Can we see these 2 organisms in hot spring systems more commonly than just, or is this just a one-time thing that we saw in Old Faithful? And so in order to get at that question, I used that matrix of hot springs that we sampled for the data that Dan was talking about for that project. And like he was saying, we extensively sampled. You can see that we have 82 hot springs that we sampled between Yellowstone National Park and Iceland. And you can see that we have a really wide range of temperature and pH that we sampled. And so again, in order to figure out who was present, we took these sediment samples. I performed DNA extractions on all of them. We had that DNA sequenced, and then that led us to answer the question of who is present and what are they capable of doing metabolically. And so from that, we actually learned that Aquifocales is extremely widely distributed in terrestrial hot spring environments. But at this point, you're probably like, who the heck is Aquifocales? Because I have not talked about that at all. So before I talk about this data more, I want to introduce you to my favorite organism, Aquifocales, because I've been working on it for the last 4 years, so I might be a little bit biased. But one really, really cool thing about these organisms is that they form these hair-like structures in hot springs. And so you can see from these videos, these organisms produce so much biomass that they're forming these macroscopic biofilms that are just kind of flowing with the water. All of these like organisms, all of this here in these pictures, all of this white stuff in this picture, that's all biomass. And most of that is from this Aquifocales organism. And so they're extremely good at living in these extreme temperatures. And so one thing about Aquifocales is that they also span an extremely wide temperature and pH distribution. And there's 4 genera of Aquifocales that are most commonly found in terrestrial hot spring environments. And so the first one that I'm going to talk about is Thermocrinus, which should be familiar because I mentioned that that's what we found in Old Faithful geyser. You can see that Thermocrinus spans and can live all the way down to pH 5 and all the way up to pH 9. And it can also live in a really wide temperature distribution. So this organism is really flexible metabolically and tends to dominate any environment that it's found in. So you can see that most of these circles are extremely large, indicating that they make up a really large portion of the community where we find them. The next organism is Sulfuri hydrogenibium. And so that organism dominates a little bit more of a moderately acidic pH range, ranging from like pH 5 to pH 7, but tends to be found in a little bit of a lower temperature range than Thermocrinus is found. Next, we have Hydrogenobacter, which is actually not as commonly found as the other members of Aquifocales, but that also is found in around pH 4 to pH 7. And lastly, we have Hydrogenobaculum. This organism is dominating that more acidic pH realm and tends to be found in lower temperatures than Thermocrinus as well. And so now that we know a little bit more about Aquifocales, I can come back to this data and show that we found Aquifocales in 49 of the 82 hot springs that we sampled. And so that's like an extremely large percentage of hot springs that have these Aquifocales organisms within them. The other question that we wanted to address was, what is the distribution of thermos in these hot springs? And so thermos was found in 26 of the 82 hot springs that we sampled. And interestingly, Aquifocales and thermos were found together in 23 of the 26 hot springs that contain thermos. And so that should really start to kind of paint the picture that Aquifocales and thermos, there must be something going on between these two organisms because they're found so commonly together when thermos is present. Another thing that I want to kind of highlight about this data here is that thermos tends to grow mainly in this pH realm. And so that's why we're not seeing any thermos in the more acidic pH springs. But the other thing that I want to note is that, like Dan mentioned in his presentation, is that these hot springs are generally fairly nutrient limited. And so we know that nutrient availability is extremely low. So here on the left-hand side, I'm showing a plot that has dissolved organic carbon plotted, and each of these little dots represents a hot spring. And so you can see after above like pH 5.5, that DOC generally is a lot lower than it is in the acidic hot springs that we have sampled. Additionally, when we look over here at ammonium, you can see that above pH 6, it's almost below the limit of detection, right around 0. And so that to us indicates that there is not a lot of organic carbon or a lot of nitrogen available within these systems. And that kind of further addresses the need for Thermos to be getting its organic carbon and nitrogen from some source. And we think that that's likely the Aquifales. And so with that, I wanted to ask the question if we can actually demonstrate in a culture this cross-feeding between a member of the order Aquifales and Thermos. And so in order to do this, I set up targeted enrichment cultures in order to obtain a cross-feeding culture. And so I have that red box highlighting which pH and temperature springs I was kind of focused on, because that's where we found the highest distribution of springs that had both Aquifales and Thermos. And I ended up setting enrichment cultures up from this Flu-5 spring from Iceland. The pH that we measured was 7.6 and the temperature was around 87 degrees Celsius. And so what we do for enrichment culturing is we collect sediment and water slurries, and then we bring those back to the lab. And I use that sediment water slurry from the hot spring to inoculate the serum vial here that contains base salt media that I supplemented with CO2, oxygen, nitrogen, and hydrogen. And so once I add my slurry to this culture bottle, I incubated them at 8 degrees Celsius in order to enrich for any organisms that are able to use these metabolites that I gave them. And then after I see that I have growth, what I do is I transfer them in order to dilute out any of the organisms that are potentially just carried over and are not actively growing. And then I ended up with a gas-supported consortium of microorganisms. And so one way that we monitor our culturing is through microscopy. And so usually once I saw an increase in growth, I would transfer them to dilute out any of the organisms that I didn't want. And I actually thought, based off of the microscopy, that I had a single morphology, because you can see that they're all really uniform rods. They're short, they look almost exactly the same. But genomic sequencing actually revealed that I had a co-culture of a previously uncultured Aquifales that I'm going to refer to as Flu-5 for the rest of the presentation. And there was also a Thermos population in that co-culture, which is really exciting because that's exactly what we were hoping to isolate. And so then we continued using that genomic sequencing, and I wanted to kind of determine and confirm the metabolic capabilities that I was expecting these organisms to have. And so here I'm showing carbon and nitrogen metabolism. And for my Flu-5 culture, you can see that it encodes an autotrophic CO2 fixation cycle, the reductive TCA cycle, meaning that it is capable of taking that CO2 and fixing it to organic carbon. Then I also was able to confirm that it had a nitrogen fixation metabolism done through NIF, which you can see over here. That's the enzyme responsible for nitrogen fixation, taking that dinitrogen and fixing it into a bioavailable form of nitrogen. But when I look at Thermos, as expected, it does not encode either the carbon or nitrogen metabolisms that would allow it to kind of sustain itself. And so this suggests that Flu-5 is cross-feeding Thermos, organic carbon, and nitrogen in my culture. And so in order to kind of show this, I wanted to determine the different growth phases of my Flu-5 and Thermos cultures. And so first I started off just trying to check or trying to set up cultures for carbon cross-feeding. So in this condition, you can see I provided CO2, hydrogen, oxygen, and ammonium chloride. And so one thing that's really important in this is that ammonium chloride is a bioavailable source of nitrogen to most cells. And so they're not cross-feeding nitrogen in this, they're only cross-feeding the carbon. And based on my growth curve, you can see that these cells are extremely happy in this condition. They grew all the way up to around 7 times 10 to the 8th cells per mil, which for a thermophilic culture growing at 85 degrees Celsius is like very, very robust growth. And so these cells were happy and they're growing well. But one problem I ran into is based on microscopy, I can't determine when my Flu-5 culture is growing versus when my Thermos culture is growing. And so I had to find another method to quantify the different growth phases of both of these organisms. And so in order to do this, I turned towards quantitative PCR or qPCR. And so the workflow for this, the first thing that I did is that I designed primers that are specific to my flu5 and my thermos cultures using the genome that I sequenced. Then I extracted DNA from each time point of the growth curve that I just showed you all. And then I used that DNA in the qPCR reactions that I set up. And I used CyberGreen as my master mix. And I used a real-time PCR thermocycler. And one thing that's important is a CyberGreen, because this is what actually allows us to quantify the level of fluorescence and quantify when each individual population is growing. And so the way this works is I have the DNA that I added, and at this point the DNA is double-stranded DNA. Then it goes through denaturation, where they are split into single-stranded DNA, and at this point my CyberGreen, there's a dye in there and it's unbound. But then it goes through primer annealing and extension. So this is where my the primers that I designed bind to the DNA, and they're eventually that single-stranded DNA turns into double-stranded DNA. And then that dye that was unbound while it was single-stranded DNA becomes bound and fluoresces, allowing us to determine when each individual population is increasing. And so here are the results for that qPCR. Again, this is the growth curve that I showed you all. And then on the bottom left here, I have my flu number normalized to the nanograms of DNA that I put into each of my PCR reactions. And so one thing on the biggest thing that you see here is that between 12 and 24 hours, we have an extremely significant increase in that copy number for flu5. It goes all the way from 1.4 times 10 to the third copies up to 1.2 times 10 to the sixth copies around there. And so that's a really, really strong increase. And so when I look at my thermos copy numbers normalized to nanograms of DNA, we also see a very strong increase between 12 and 24 hours, where it increases from 1.5 times 10 to the third up to a little over 7.5 times 10 to the third. One thing that I want to note here is that the scales on these are extremely different. And so although we have an increase in thermos, the increase is a lot less significant than the increase in flu5, but that's exactly what we would expect to see, because thermos is relying on flu5 in order to obtain all of the carbon and nitrogen that it needs. So we would never expect to see thermos outcompete or outgrow flu5 in this culture. The other thing that I want to point out here is that after 24 hours, my flu5 cells aren't as happy anymore, and we see based on the error bars that there's a lot more variation, and they kind of start decreasing, and that trends exactly with what we see happening in the thermos. Once flu5 stops increasing, thermos is not supported anymore by the flu5 culture. And so this really showed that the growth of thermos is dependent on that major increase in growth that we saw in our flu5 sample. And so the next question was, how does nitrogen fixation impact the patterns between flu5 and thermos when they're cross-feeding both nitrogen and carbon? And so in order to answer that question, I set up a really similar experiment where this time I gave them CO2, hydrogen, oxygen, and nitrogen instead of ammonium chloride. And nitrogen is not a bioavailable source of nitrogen. And so what needs to happen for thermos to use this nitrogen is that the flu5 needs to convert that into a form that thermos can use. And one thing that I want to note again here is that my cells per mil are a lot lower than the cells per mil in the culture with ammonium chloride, because nitrogen fixation is an extremely energetically taxing metabolism, and so we wouldn't expect the cells to grow as well when they're fixing nitrogen. But because of that, I also had to increase the amount of biomass that I was harvesting with each time point. And so the way I did it is I had an early time point, a middle time point, and a late time point that I sampled for this. And unfortunately, I'm going to have to leave you all on a cliffhanger for this data, because I am still working on processing the qPCR. And so if you're interested in knowing what happens with this, you can follow up with me, or hopefully in a few months, read the paper once it's published. hopefully. And so one other thing that I want to kind of talk about before wrapping up is the future directions of where we're going with this project. So in the next few months, like the things that I kind of want to do to finish this up is to do stable isotope probing with these experiments. And so the way stable isotope probing, or SIP, works is I have the same kind of culture setup that I've been working with my nitrogen, carbon dioxide, oxygen, and hydrogen. But one difference is that I have two different experiments that I need to set up. One is the control, where I give them just normal carbon and normal nitrogen. And the other control is where I give them labeled carbon and labeled nitrogen. And that label ends up making the DNA heavier, which allows me to separate the DNA, the heavy and the light DNA through ultracentrifugation, so that I can actually see the cross-feeding behavior between Flu-5 and Thermos to actually like confirm with numbers that they are being cross-fed. And so that's up and coming. And I know I threw a lot of information at you all, and so I want to run through some main conclusions for this work. The first thing is that we determined that Aquafocales and Thermos are found together in alkaline hot springs. We also had a gas-dependent microbial consortium that we enriched from an Icelandic hot spring that we sampled in order to probe these cross-feeding dynamics. And then based on the genomic data of that co-culture, we kind of hypothesized that Thermos is dependent on Aquafocales for carbon and nitrogen cross-feeding based on the metabolisms that both of these organisms are encoding. And lastly, preliminary qPCR experiments show that the growth of Thermos in my co-culture is dependent on an increase in Flu-5. And so with that, I'd like to thank everyone that has made this work possible and has supported me through this, and take any questions.
What you're really saying that we wouldn't be able to do PCR, molecular biology, all without thermocritus?
No. Oh, yes, yes. So, yeah, he means, so Thermos is actually where TAC came from. So TAC is an extremely important part of kind of the more like biotech application of microbiology, and allows us to do a lot of the molecular work. And so Thermos was isolated by Thomas Brock, and we found TAC, and so that kind of opened up a lot of doors for what we can do with the like biotech applications in sequencing and PCR of microbiology.
That there are other microbes that are playing a role in this in the more acidic springs?
So the acidic springs, like Dan was saying, is dominated by sulfolobales, which are also autotrophic organisms. And so there's like a kind of, every hot spring community will most likely have kind of a similar structure where there's like an autotroph that's dominant that is cross-feeding heterotrophic populations, because the heterotrophs in hot springs are relying on those autotrophs for carbon, for nitrogen. One thing that I do want to point out is that acidic hot springs do have more nutrients available. So they do have like higher levels of that dissolved organic carbon and different nitrogen species. And so there might be less of a dependence on those autotrophic communities for cross-feeding for heterotrophic populations in the acidic hot springs. Yep, Anna.
Is the thought that in those 3 hot springs where you found Thermos but not Aquafocales, that they're probably cross-feeding off of some other population?
Yeah, that's actually one of the things that I want to do is look into what's going on in those communities. But I would expect there's like a different dominant autotrophic population in those hot springs that just isn't a member of the order Aquafocales. I don't know who it is or why it isn't Aquafocales because I haven't had time to look into that, but that is definitely something that I plan on doing. Thank you, everyone.